Hello dear students, welcome back. This is Dr. Hadi here. Finally, we got this topic, lac operon. Finally, I'm going to discuss this topic of lac operon after your repeated request. So let's discuss it in detail, and I would request you to watch this video from the beginning till end, and I guarantee you will understand in my simple language, simple English language. The concept of operon, which is also called as lac operon, it actually related to the regulation of gene expression. And you know, we have started the chapter of regulation of gene expression. So this topic belongs to that chapter, regulation of gene expression. What is operon? The definition is here. It is mentioned here: a coordinated unit of genetic expression. And the two scientists, Jacob and Monod, they discovered this topic first in 1961. This is 2020. This definition is very difficult. You cannot understand this definition. Let me explain you. As we have discussed earlier, that in the past the scientists think that in the DNA there is one gene, and that one gene is responsible for one enzyme. This was the concept. Later on, we came to know that there is not a single gene on the DNA. Let's suppose this is your DNA. There is not a single gene that is involved for one particular enzyme. But now, there are so many genes. They work together. There are so many genes, a group of genes on the DNA. They work together. So that's why I use the word coordinated. These genes work together to start a complete process, just like the metabolism of glucose or the metabolism of some other biological molecule. Like in this topic, we will discuss the topic in the prokaryotes, in the bacteria, lactose metabolism. Lactose is a disaccharide, and its metabolism require at least two enzymes. Now, these two enzymes and the whole process will be carried out by the help of some genes. These are mentioned on the whiteboard. So this is gene 1, 2, 3, 4, 5. At least 5 genes are there, and these 5 genes will work together to support that metabolic process. And I would like to give you some information about this operon. This is the operon, right? This first of all, for some students it is just a diagram, but it is actually the DNA. This is actually your DNA. It's a long strand of DNA, but this is one segment of the DNA. This is one segment of the DNA, and that segment, only that segment, is involved in the metabolism of the lactose. Lactose is a disaccharide. I repeat once again, and in shortcut we write it LAC, lac operon. And this small segment is the DNA. The small segment of the DNA is called as operon, right? This is the operon. And this whole segment is not responsible for the synthesis of just one enzyme. It is responsible for the synthesis of more than one enzyme, plus this segment is also involved in the regulation of these genes. Means either you want to stop the process or you want to start the process. So all of these regulatory activities, expressing activities are just packed together. They're just packed together in just one segment of the DNA. Everything is there. With just one button, you push it on, the enzyme synthesis start, and you with another button, you just push it off, and the process will be stopped. Here and that one segment, The first part is the regulator. The first part is the regulator gene, right? This is not a site, is a gene, is one gene called the regulator gene, and we write it with I. What this regulator gene does, the regulator gene synthesizes a protein, a special protein, and this is a kind of constitutive gene that I have just discussed in my previous video. This is a kind of constitutive gene, and it means it is required for all the time as far as when the lactose is not present in the bacterial cell. This gene will be working all the time until unless when the lactose does not enter the cell. So this is the first gene called regulator gene. What it synthesizes? It transcribes a messenger RNA, and with the help of this messenger RNA, it produces a protein called repressor subunit. And this is actually a protein, right? This is a protein, and it consists of 4 parts. 1, 2, 3, 4. 4 parts. This is just one protein, and this whole protein is called a repressor. It is a kind of signaling molecule. It is a kind of signaling molecule whether to start the lactose metabolism or to stop the lactose metabolism. It all depend on that molecule. This is just a tetramer. Okay, here you can write tetramer. 4 units are there, and this tetramer, which is called repressor subunit, it come to us because of the messenger RNA, and messenger RNA has been produced by the regulator gene, because this is the DNA, and DNA can copy all of its information always in the form of messenger RNA. So whenever an information come from the DNA, it will not come directly. The information from the DNA will always come in the form of messenger RNA. Remember, if this is the DNA, so this is the RNA, and it is one of the type of the RNA, messenger RNA. Messenger RNA will always carry a message. So this is the tetramer, a repressor molecule. What this repressor molecule does, as far as the lactose is not entered into the bacterial cell. Remember, this topic is also applied for the eukaryotic cell as well, but today we will discuss this process in the prokaryotic cell, in the E. coli. Bacterial name is E. coli. This tetramer, which is kind of a repressor molecule, it will come and will attach to another place, another site on that DNA called the operator gene. So it is not a site, it is another gene, operator gene. And this is a second gene, we write it O, operator gene. When this protein is attached, let's suppose the protein is arrived here, the protein is arrived. So this protein which is arrived here, and it is attached with the operator gene. Now what will happen? Because of its particular shape, it will block that promoter site. It will block that promoter site. And do you know what happens on the promoter site? A promoter site is not a gene. It is, of course, just a site for the attachment of enzyme called polymerase. So there is an enzyme. Let me write here. This is an enzyme. It should be written as E, enzyme E, and it is called as RNA polymerase. This is called as RNA polymerase. Okay, this is an enzyme. And you know, RNA polymerase is always required to synthesize a messenger RNA. You know, RNA, what type of RNA, whether it is messenger RNA, transfer RNA, any type of RNA, these are always came to us from the DNA. They are synthesized from the DNA, and they need enzyme, the RNA polymerase. So this RNA polymerase E will come here, but this enzyme will not be able to attach on the promoter site. Why? Because of that repressor molecule. This repressor molecule will not let the enzyme to attach on the promoter site until and unless this molecule leave that operator gene. So up till now, as far as the lactose has not entered into the bacterial cell, this whole DNA piece will not start the synthesis of some enzyme. Means there will be no metabolism of the lactose. Lactose is not present, so there is no need of metabolism. But when in the bacterial cell, let's suppose this is the bacterial cell, right? This is the bacterial cell now. This is bacterial cell. When that lactose is a disaccharide, okay, I repeat, a galactose and a glucose, combination of a galactose and a glucose is called lactose. This lactose, when entered in the bacterial cell, it induces some changes in that cell, right? How? Now see, now let's suppose this is the lactose, okay, let's suppose this is the lactose molecule, and I will write it as L, lactose molecule L. When the lactose reaches in the cell, this lactose has a very high affinity with that repressor molecule. These two are each other's lover. They love each other too much. So whenever the lactose reaches and the oppressor molecule will leave its place, now the oppressor will leave this place. Why? Because some kind of changes will take place. Changes will take place in the repressor. Why? Because that L, lactose, will bind to it. That repressor molecule will not leave the operator gene. Once the lactose is attached, let's suppose this is your repressor molecule and this is lactose. When the lactose is attached with the repressor molecule, it brings some changes in the repressor molecule. Because of these changes, the repressor molecule will leave the surface. That operator gene will leave. And here your lactose is now attached with what? With that repressor molecule, as 4 segments repressor molecule. Now finally the operator gene is free. Now the site for the promoter site is also now available. Now what will happen? That enzyme RNA polymerase will attach at the promoter site. Now this is the enzyme, it will attach at the promoter site. Now as the enzyme is attached at the promoter site, now what will happen? I will erase this, okay, this messenger RNA, I will erase this. Now what will happen? Because of the RNA polymerase, we will get the RNA. The RNA means these are 3 genes, Z, Y, and A. Okay, these 3 genes are called as structural genes. These genes are called as structural genes, and these genes will copy their messenger RNA once the polymerase attaches here at the promoter site. So now this region which is called Z gene, it will always produce beta galactosidase, of course by the help of its messenger RNA. So if I just write this enzyme here, beta galactosidase, beta galactosidase. It is an enzyme. And how this enzyme will be formed? When that gene Z transcribe a messenger RNA. Messenger RNA. As messenger RNA will come. And one thing more, 3 genes, 1 single messenger RNA. This is called cisternal messenger RNA. Usually there must be 1 messenger RNA for that gene, another messenger RNA for that gene, another messenger RNA for the 3rd gene. But no, this time only 1 messenger RNA will be there. Only 1 messenger RNA will be there. Only 1 messenger RNA. Like this is only 1 messenger RNA, and this messenger RNA contains the information of Z gene, the information of Y gene, the information of the A gene. All these 3 genes information are placed in this 1 single RNA. Now when this RNA is expressed, of course this segment will give us beta-galactosidase enzyme, and this segment when expressed, of course when expressed, this segment will give us permease enzyme, and this 3rd segment will give us another enzyme called acetylase. The 3 enzymes will be formed, and good news is that these 3 enzymes are involved in the metabolism of the lactose. How? The enzyme beta-galactosidase will break down, hydrolyze the lactose. This is your lactose, and it will hydrolyze it into 2 pieces, that is glucose and galactose. Hydrolyze it. The permease is another enzyme. It will help the lactose to enter into the cell, right? And the 3rd enzyme, acetylase, its function has not yet been cleared. Fine. So this function is not cleared yet. However, these 2 enzymes, permease and galactosidase, these 2 enzymes will start the metabolism of the lactose. Now, starting point, lactose metabolism, need enzyme, and these 2 enzymes will start the metabolism of the lactose. And now you just think for a while, we have one process. What is one process? The lactose metabolism is just one process, and for this one process we don't have one single gene, but we have a segment of the DNA that contain many genes, and these all genes work together to promote one metabolism. These all genes work together to start the process or to stop the process. So remember, in the beginning, when the repressor molecule was attached here at the operator site, when the repressor was here and there was no enzyme polymerase on the promoter site, at this stage the whole process was stopped. This is called as repression. This is called as repression. This is one topic, repression. So what is mean by repression? I can write here. Repression means regulator gene I will make a messenger RNA, and that messenger RNA will give us a molecule called as repressor molecule. That repressor molecule will attach at which point? Will attach at the operator gene. What will happen? Once at the operator gene, the promoter site is blocked. The promoter site is blocked. When the promoter site is blocked, no polymerase enzyme attach. Okay, when no polymerase enzyme attach, these 3 genes will not be expressed. these three genes will not be expressed into the messenger RNA, and because of this messenger RNA, no enzyme will be formed and the process will be stopped because no lactose is there. Second heading is depression. And what is depression? It is the opposite of repression. In depression, a lactose molecule will come. That is why lactose molecule is called inducer. Because of the lactose molecule, that repressor molecule will attach with the lactose, bring some changes into the repressor molecule. As a result, it will remove itself from the operator gene. Lactose inducer, repressor inactivated, mean it will leave the operator gene, no binding to operator gene, and then what will happen? Polymerase attached to the promoter site. So polymerase here attached at the promoter site, then what happen? Transcription start. Means the messenger RNA from these gene will be made. There should be 3 RNAs, but in this case only one single RNA is formed, and this one single RNA will code the 3 enzyme, that is galactosidase, permease and acetylase. And galactosidase will hydrolyze the lactose, permease will help in bringing the galactose inside the cell, and acetylase has no function. So we saw what a great check and balance in the metabolism of the lactose. Lactose. When lactose is not present, absent, then this will be called as repression, and the whole detail of the repression is here mentioned. And when a lactose enter in the cell, then another process will start called as depression. Repression, depression. Depression means lactose metabolism start. Lactose metabolism start. Repression means lactose metabolism stop. Repression and depression. So this was a topic. I tried my level best to express my words, my ideas to you people, and of course I'm expecting that you will get this topic, and I'm also expecting that you have enjoyed watching that video. Thanks for watching once again. See you in the next video. Bye bye. Tata.